President of Phelan-McDermid Syndrome Foundation. Thank you so much for joining us. We are excited to introduce the Phelan-McDermid Clinical Research Study, Developmental Synaptopathies Consortium. This is one of many exciting initiatives the Foundation is supporting. So in order to help fund projects like this, we rely on wonderful events put on by our families and our fantastic donors. And let me just do a little plug here. One of our biggest fundraisers is September 1st, called the Giving Challenge. We would so appreciate everyone participating. Please check the website and Facebook for information. Thanks so much. We will try to answer your questions today if we have time, but if we don't, we will follow up with the questions. And Geraldine has a slide to tell you how to do the questions. I'd like now to introduce Geraldine, who's instrumental in organizing this research effort.
Thanks, Sue. So before we get started, I just want to also make a quick plug for the iPad raffle that we have going on right now as part of the Phelan-McDermid Syndrome Data Network. If you are interested, you can participate in CareSync, which is a service which obtains all of your child's medical records and collects them and maintains them in one centralized place. All of the data that's collected will be part of the Phelan-McDermid Syndrome Data Network, so it's really easy and convenient. And you are also entered into the iPad raffle. So if you want to do it and you want to take part, just email Jackie at pmsf.org and she will help you get organized. But you have to do it before August 21st in order to be eligible for the drawing. Okay, so we're really excited to present to you today a webinar about a very important and brand new clinical research consortium called the Developmental Synaptopathies Consortium. This is a really special time for our disease. If you've listened to me talk before, you've heard me talk a lot about mouse models and induced pluripotent stem cells. And because of the amazing work that's been done by the bench scientists, we're in the unusual situation that really the biggest gap area right now is clinical research. That is knowledge about what is going on with the human patients, with kind of the natural history of the disease, with the identification of biomarkers that are going to be very important for clinical trials. And so this new clinical research consortium will address many of those research gaps. So I'm delighted to introduce you to two doctors and researchers, Dr. Sahin and Dr. Kolevzon. Dr. Sahin is he's a professor of neurology at Harvard Medical School, and he is the director of the Translational Neuroscience Center at Boston Children's Hospital. And he is a real expert, just a real leader in the field of tuberous sclerosis. TSC is a disease that's related to Phelan-McDermid syndrome. It also causes autism, epilepsy, and intellectual disability. He is an investigator with the Autism Centers of Excellence, which is another network of clinical research sites where they are looking at certain high priority autism research topics. And he is also the principal investigator of this new clinical research network involving Phelan-McDermid syndrome, the Developmental Synaptopathies Consortium. He's also a practicing child neurologist. Our other speaker today is Dr. Kolevzon, and most of you may already know him. He's participated in many of our webinars in the past and our family conferences. He's an associate professor of psychiatry at Mount Sinai. He's the clinical director of the Seaver Autism Center. He is an expert in the diagnosis and treatment of autism and related neurodevelopmental disorders. His area of focus has been developing new pharmacological interventions and identifying biomarkers and studying objective outcome measures. And he has been just a huge part of this project, of the Developmental Synaptopathies project. He developed the PMS phenotyping protocol that will be used by all of the sites. So I'm pleased to turn it over to Dr. Sahin right now. And I'm going to, Dr. Sahin, do you have the slides, or should I just transfer controls over to Dr. Kolevzon and let him walk through the slides?
I think Alex has the slides, right, Alex?
Yeah. So,
okay.
Mustafa, just let me know when you want me to advance the slide.
Okay. Let's go to the first slide, I guess.
Okay. So, Geraldine, you're going to give it over to me?
Yep. You should have it in just a second.
How's that?
That's great. Thank you.
There you
Well, thank you, everyone. I also want to thank the Phelan-McDermid Foundation for really making the grant possible for us to be able to apply to. We really wouldn't have been able to get this grant funded without their help from the NIH. The Developmental Synaptopathies Consortium is one of the 22 consortia that's funded by the NIH as one of their rare disease clinical research networks. And it's the only consortia that's really focused on diseases that are associated with autism and intellectual disability. So it's really unique in this context. Can we go to the next slide, Alex? So the name of the consortium is Developmental Synaptopathies, and most of you probably are familiar with the word development, but synaptopathy may be a new word for some of you, and it really means a dysfunction of the synapse. And synapse is where the two nerve cells connect to each other and communicate. And we believe that this type of abnormality at the synapse is underlying many of the abnormalities that we're seeing in patients affected with intellectual disability and autism these days. So different synapses in different parts of the brain may result in dysfunctions that are relevant to communication, social interactions, higher level functioning and processing in the brain. And all of the diseases that we're studying in this consortium have some amount of or some type of synaptopathy. Next slide, please. So as a part of the rare disease consortium, we're going to focus on 3 rare diseases. They all share the feature that they all present neurological abnormalities, and in our understanding at the cellular level, abnormalities at the synapse. They all have high incidence of autism spectrum disorder. And being able to look at these 3 diseases comparatively will allow us to identify common features that might be relevant to autism and intellectual disability. So we're going to actually use the fact that these 3 diseases share certain features to try to identify what are really the core circuit abnormalities and cell pathway abnormalities in autism spectrum disorder and intellectual disability. Next slide, please. So one thing that's becoming really clear in autism is that it's a very heterogeneous disease. And what I mean by heterogeneous is that there are probably about 500 to 1,000 different genes that give you susceptibility to developing autism. Now, this is helpful in terms of identifying the cause of autism, but when it comes to treatments, it actually becomes an impediment. Are you going to develop a different drug for every gene that's associated with autism? That's probably not a very likely scenario. It's also equally unlikely that there will be one treatment, one intervention, one drug that's going to be broad spectrum, such that you can treat all types of gene mutations that are associated with autism in the same way. What might be more likely is that there might be drugs that work for subcategories of autism or genes that are similar to each other that are following in the same pathway. Now, I should say this is probably not very much different than how doctors who take care of infectious diseases these days take care of patients. For let's say a bacterial pneumonia, there's not just one antibiotic that takes care of all pneumonias. And equally, we don't have one antibiotic for every different microbe that's a different strain of microbe or different strain of bacteria. But there are some antibiotics, for instance, that work for certain types of bacteria, and there are other antibiotics that work for other groups of bacteria. And this is sort of analogous in the way we're starting to think about brain disorders as well. So we're hoping that some diseases will fall into a subcategory, and we may be able to treat those diseases with the same interventions. Next slide, please. So Developmental Synaptopathies Consortium focuses on 3 diseases, as I mentioned earlier, and these 3 diseases are Tuberous sclerosis complex, PTEN-associated hamartoma syndrome, and SHANK3 mutations and Phelan-McDermid syndrome. The consortium is made up of 10 different centers. They are listed at the bottom of the slide. I'll read them. Boston Children's, National Institute of Health, University of Texas at Houston, University of Alabama at Birmingham, Stanford University, Icahn School of Medicine at Mount Sinai, Rice University, University of Texas Southwestern, University of California in Los Angeles, and Cleveland Clinic. There are a number of reasons why we chose these institutions. First and foremost, they have large populations of patients that they already follow with each of the particular diseases. And second, they are geographically distributed, so we can provide the widest coverage in terms of recruiting new patients and making the trips to the centers as easy as possible on the subject. Next slide, please. The long-term goal of the consortium is to characterize these three genetic disorders, which all, as I mentioned earlier, all have a high penetrance of autism spectrum intellectual disability, to shed light on the molecular pathways and targets relevant to autism and intellectual disability. In the short term, what we want to achieve is to characterize the neurodevelopmental phenotype of these three groups of patients longitudinally. So we'll be performing natural history studies in relatively large sets of patients with each of these genetic disorders. And second goal is to identify biomarkers that predict risk for disease severity and progress. And that second one is really directly relevant to hopefully finding treatments for these diseases, because we all believe these days that biomarkers will accelerate treatment trials going forward. Next slide, please. So the three studies that have actually already gone through the IRB and are open for enrollment are listed here, and they each represent one disease. The first one is about tuberous sclerosis. The second one is about PTEN mutations, and the third one is on Phelan-McDermid syndrome. And Alex will talk more deeply about the third project. But what's unique and what really leverages the fact that we have a consortium is that all three projects share as much as possible common neuropsychological tests and common imaging platforms, so that we can actually compare those three groups of patients across the three diseases as well. Next slide. So I'm not going to spend too much time on this, but the goals of the Tuberous Sclerosis project is to characterize the developmental phenotype of autism and intellectual disability in children with tuberous sclerosis, to identify biomarkers, especially using MRI imaging, and third is to establish infrastructure for collecting biospecimens. So this could be brain tissue or blood or skin biopsies from patients with tuberous sclerosis and their family members. The third goal is really for understanding the disease more deeply at the cellular level, and having access to human specimens is a major obstacle in many of these rare diseases. Next slide. Similarly, in PTEN, we're going to look at the medical, behavioral, and cognitive differences between PTEN, ASD, and other groups. So in that case, we have several groups, some patients with autism spectrum disorder and macrocephaly, but no PTEN mutations, and then others with PTEN mutations with autism spectrum disorder. And we're going to compare those two groups. We're also going to look at neural systems and molecular biomarkers, similar to the TSC project, in the PTEN patients. And then the final aim, similar to TSC, is again to have a biorepository collecting samples from patients affected with PTEN and autism. Next slide. And just for symmetry, the Phelan-McDermid project is very similar to the other two projects. The first aim is to look at the natural history of the syndrome using longitudinal assessments. Second aim is to look at neuroimaging as a biomarker. And the third aim, which is slightly different from the other groups, is to look at genetic practice, because this is a syndrome where the genetic mutation may actually differ from patient to patient. Next slide, please.
That's it.
All right, I'll stop there then. Back to Alex.
Okay, thanks, Mustafa. Thanks for that terrific overview. And Geraldine and Sue, thanks for inviting me. It's always nice to talk to this group. I'm looking forward to kind of giving a somewhat detailed overview of the protocol and also to introduce you to our sites. So as Mustafa noted, we have a whole variety of aims. I think taking a step back, for us, the main goal here is really to set the stage for developing clinical trials. We want to learn as much as we possibly can about the phenotype. We want to characterize Phelan-McDermid syndrome from a medical perspective, a cognitive perspective, and also using a whole host of behavioral measures. Really important for developing clinical trials. We want to know, how does this syndrome change over time? When you pick outcomes in clinical trials, you want to make sure you don't pick a moving target. You want to make sure you pick something that you think is likely to improve. And so this is a really critical piece of this project. Also, as Mustafa mentioned, we want to use neuroimaging, including diffusion tensor imaging, to look at the myelination integrity. And we're very interested in how the phenotype changes according to how large the deletion is, where the deletion is, and how many other genes are affected. So to accomplish these aims, we've set up 6 sites: Mount Sinai, Rush University with Liz Berry-Crevice, and Latha Surya. And Latha actually came from the Mount Sinai site, and is someone who's seen probably 30 or 40 patients. Boston Children's, Mustafa has just assumed the investigator role there. Audrey Thurm at the National Institute of Health, Craig Powell at UT Southwestern, and John Bernstein at Stanford. So among these sites, we're hoping to recruit at a minimum 90 patients and closer to 100, we're hoping, with these 2 additional sites we just added with the support from the Phelan-McDermid Syndrome Foundation, UT Southwestern, and Stanford. So we're going to be doing some very kind of deep characterization. Everybody that comes through will be getting physical exams, neurological exams, clinical genetics evaluations looking at dysmorphology, medical and psychiatric history. We're going to be looking at the heart, at the kidneys. We're going to be doing a lot of laboratory blood work. We hope everybody that comes through will either have had or will get EEGs, and then a number of other kind of routine measures. In terms of the more behavioral and cognitive phenotyping, we're going to be looking at global cognitive ability, adaptive behavior, language, motor functioning. We're very interested in autism symptoms and really what the prevalence of autism is within Phelan-McDermid syndrome, and even more importantly, what the validity of our current scales are and the instruments that we use, and how perhaps in the future we can refine some of these instruments to be more relevant and more specific to Phelan-McDermid syndrome. And then we're also interested in sort of the broad neurobehavioral phenotype, all kinds of aberrant behaviors, repetitive behaviors, all the types of things that we know happen with our kids, but really the prevalence hasn't been really well established. So I wanted just to go through just quickly some of the sites and introduce you to some of the main players. So the Mount Sinai site, I know a lot of you are already familiar with. We've just added a clinical geneticist, Raimundo Lozano, and a developmental pediatrician to our group, Christina Farrell. The key player here is Emily Forey, who's our research coordinator and the person that will be coordinating all of our visits. And so if you need to reach her, this is an information, and I'll show this again, and I think Geraldine will also show it again. But this is a really big, terrific team of child psychiatry, developmental psychology, clinical psychology, peds neurology, Dr. Frank, and so on. Latha and Liz Berry-Crevice. Here's Latha. Can you guys see my pointer?
Yep.
Oh, good. And here's Liz Berry-Crevice. This is Jane, their research coordinator. They're at Rush in Chicago. These guys, as of next, I guess in November now, are going to actually establish a clinic. So in addition to seeing kids in the context of the RDCRN protocol, they're also going to be available to treat kids on an ongoing basis and of all ages. This is the Boston Children's Group under Mustafa's leadership. This is Rania, their fearless coordinator. And I guess Megan just joined, right?
Right.
Yeah. And this is where these guys are located. We had a really terrific launch meeting there in the spring, and all of us got together and met each other and had a really productive meeting. At NIH, it's led by Audrey Thurm, along with Sue Sweeto, and I believe this is their nurse practitioner, Priscilla. This is their research coordinator, Maggie. This is a wonderful site, really, really well established to support families. They're going to bring a lot of people through. They've already started, actually. And then at UT Southwestern, which is run by Craig Powell's group, I don't know who all these players are, but it's obviously a very large group. They're focused on autism. They're focused on a lot of different developmental disabilities. They have a ton of disciplines within their group: pediatric neurology, psychiatry, psychology, speech. They do ABA. They have access to really terrific consultants. This is a fantastic group. And Craig actually gave me this slide, and he left off the most important picture, which is him. This is Craig, the PI. I'm supposed to get a laugh. Come on. No laugh. Thank you.
I'm muted.
Okay. John Bernstein's at Stanford. Here's John. This is the neuropsychologist. They have a coordinator named Julia who's not pictured. This is obviously a beautiful place. And this is one of the new members of our consortium. So moving on to the second aim, again, this idea of natural history, we really don't know what the natural history is. We know that the phenotype, that the clinical characteristics of the syndrome change over time. We know that there's different time points that increase the risk of seizures. We know that there's some kids who have regression, some kids who have different kinds of mood cycling presentations, and we're interested in trying to capture that and figure out what the real risk periods are. So toward that aim, we're going to be seeing kids at 3 different time points. So there'll be a baseline assessment, then we're going to see them at 1-year and 2-year follow-up. So there'll be 3 time points over the 3 years, and we're going to be doing a very, very similar assessment each time, really trying to capture change over time. In terms of the biomarker aims, we're going to be using magnetic resonance imaging, including diffusion tensor imaging. So diffusion tensor imaging is a measure essentially of the myelination integrity of the brain. So this is the way that the brain sort of conducts signals is very much dependent on myelin. So we're looking at both at structural brain abnormalities, but also on myelination. And so we've done a little bit of work in this area up until now, and I think I've scanned about 11 or 12 kids with Phelan-McDermid syndrome and compared them to kids who have autism due to some unknown underlying genetic etiology. And what we found is actually the integrity of the myelination. There's sort of a delayed myelination that's relatively diffuse across the entire brain. But we're very interested in this as a measure and some of the other structural changes that we hope to find in order to both predict disease progression, or at least to understand disease progression better, but then also perhaps identify specific markers that might be able to predict treatment response. And then our last aim is another important one in our view, and it's really to understand why so many of these kids, and this is some of the really cute kids that we've seen about 50 kids up until now, and really to try to understand why so many of these kids look so different and why they present so differently. Not just at different time points in their life, but even in a snapshot of time. If you take 5-year-old kids, 10 or 15 of them, they could all have very different features, from gastrointestinal problems to cardiac problems to kidney problems to seizures and so on. So there's been a lot of work in the field trying to figure out whether the size of the deletion, so in other words, the number of genes involved in the syndrome, including SHANK3, have an effect on the severity of the syndrome. And there's been a lot of conflicting evidence, and I think most of that's because the sample sizes have been very small. Ours included looked at the size of the deletion compared to a number of different features. And we did find that the total number of medical problems, the total number of dysmorphic features, and even some of the qualitative abnormalities in social interaction all seem to get worse or were at least more severe the larger the deletion size. And of course that makes sense because there are more genes involved. In the registry, along with Catalina Betancourt, there's been a fair amount of work looking at some of the other phenotypic changes. And here you can see that with kids who have very small deletions where only a few number of genes are affected, including SHANK3, you actually very rarely see congenital heart defects. But then as you get into the larger deletions, it does seem as if these kids are much more likely to have heart problems. And I think that's going to be true across a variety of different problems. And we'd like to know that in advance. We'd like to know even perhaps within this region, are there specific genes that may contribute to the congenital heart defects, that may contribute to the seizures. It would give us a lot of information to give to families about what to predict and what to monitor more closely over time. So towards that aim, we're going to be doing whole exome sequencing, looking at the entire exome, and seeing whether there are different genes within the exome among all of our genes that perhaps contribute to the phenotype in Phelan-McDermid syndrome. And so this is across the sites. This is a study contact information. I apologize, I don't have the Stanford information up there, but we'll get that to you as soon as it's available.
Alex, I uploaded a file. It's in the handout section of the control panel, and it does have the contact information for Stanford.
Oh, great.
So for everybody listening in, just look on that side panel, that control panel, look under handouts, and you should find a PowerPoint slide there that includes all of this information.
Okay, that is great, and obviously we'll give people access to these slides, and maybe we can even post this webinar online. And with that, I will end, and note, by the way, Geraldine, that it is exactly 25 minutes after the hour.
Very good, Alex. Terrific. Okay, let me just put my screen back up here. So tell us, what are the age ranges that are included in this study?
So thank you for bringing that up. I meant to mention that. So for this particular study, we're going from 3 to 21. Although I can say that many of the sites have lots of ongoing studies that are going on that we can provide information about that will be relevant to kids both below 3 and over 21, our site included. So I think as it relates specifically to this study, it's 3 to 21, but that shouldn't deter you from contacting your closest site. I have a couple things actually on that point that I'll mention. So if you're interested in participating in Phelan-McDermid research in general, or interested in getting in contact with any of these sites that are convenient, you should contact them regardless of age, because we don't know exactly how much they have going on beyond this particular study. And we will know soon because we're going to distribute that. The other thing I wanted to mention was that we have travel funds available through the Phelan-McDermid Syndrome Foundation to provide families with some help to get to these various sites. I think we're going to encourage people to go to their closest site. I think that's probably most convenient, especially because we're talking about not just one time point, but 3 time points over 3 years. But to speak with the coordinators at the specific site so that we can help you with travel funds.
Great. I'm glad you mentioned that. That's actually one of the ways that the foundation is supporting this project. We provided the funds to support patient travel. We also provide funds for the inclusion of Stanford and UT Southwestern in this consortium because we wanted sites all across the country and wanted to make sure that there wouldn't be a huge travel burden on families. So we're delighted that we've now got this wonderful network of sites, hopefully conveniently located for families across the U.S. Can international families participate also?
Yeah, there's no exclusion. It's just that's a logistical issue. I think we have a sort of a finite amount of travel funds available, but we're not going to discriminate against international participants.
Okay. Are there any phenotypes that would cause them to be excluded from the study?
Sorry, I just wanted Mustafa had a point that was relevant.
Oh, I'm sorry, Mustafa.
Based upon the analysis we're doing, we can only take English-speaking patients just for the requirements of keeping everything standardized. So I just want to bring that up in terms of international patients.
Okay,
right,
that's a great point. Yeah. Okay. Are there any specific phenotypes that would cause somebody to be excluded from the study?
No, if you've got a deletion or a mutation in your SHANK3 gene and you are between 3 and 21 years old at the time of enrollment, then no, there are no other exclusions.
Okay. Somebody asked, what is a natural history and what do you mean by longitudinal?
So those are actually 2 ways of saying the same thing. So longitudinal, over time, in a prospective way, collecting information about the characteristics of the syndrome. And if you do that, at a certain time point, and then 1 year later, and 2 years later, and even 5 years later, you can get a sense of how symptoms change over time. That's what's called a natural history. And again, sometimes you do studies where you look at the effect of a specific intervention. So you'll do an assessment at baseline, and you'll do assessment 12 weeks or 6 months later to try to capture what the effect of an intervention was. But here we're trying to capture what the effect of time is. Obviously the interventions will be involved, but what does the syndrome look like at different stages of development, and how does the syndrome change over time? Those are the natural history questions.
Okay, we have a couple more questions, or several more. What can people with mosaic deletions or ring 22 participate?
Yes, absolutely.
How many days will our children be required to be on site for the study per visit?
Right, that's a great question, and I think it's going to vary a little bit according to the sites. And so that's a really good question to ask the coordinators. I would say at our site, the assessment takes 2 or 3 days. You really can't expose kids to that much testing and expect to get a meaningful evaluation. We want to try to get the best that we can out of the kids, but that does mean that they're kind of hopefully well rested, well fed, and not exposed to too many hours at a time of testing. Some of the testing is done with parent reports. Some of it's just medical testing where they just have to lie on the table, for example. But it will take 2 or 3 days, I would say.
Okay. We've gotten a few questions about the age range, and let me just sort of combine a couple of questions here. If my child is outside of the age range, like older than the age range, should I just go ahead and contact the closest site and see if or how my child can participate in some type of research study going on at that site?
Yeah, absolutely. And I think I would say wait another week before you do that, because we're about to distribute all the different studies that are going on at our various sites. Because I know, for example, we're happy to see, and we have studies going on at virtually every age. Stanford has studies going on at every age for kids with sleep issues. I think UT Southwestern has studies going on at every age using different kinds of outcome measures. So there are a lot of studies going on. And yes, you should contact your local site.
Okay. Are you only trying to recruit families that live near the listed sites?
No, no. We chose these sites in part because they represented many different geographic areas across the U.S. We'd like to capture, as many families as we possibly can. So there's a certain feasibility in terms of the cost. Obviously, the further you travel, the more expensive it potentially could be. But no, we're not discriminating based on geographic location.
Right.
And we want to be as representative, the sample to be as representative as possible. So just recruiting some subjects around the institutions would not achieve that. So I think the funding that the Phelan-McDermid Syndrome Foundation has given us in terms of travel funds will really enable us to get the most representative sample for these studies.
Right. Right, and that's probably a great place to stop. It is just after 11:30 or 11:30 Central Time, my time, and we probably need to go ahead and wrap up the webinar. But I'm glad we get to end on the travel funds. This is provided by the foundation, and the foundation, the reason or the way that we're able to do all of this, support these kinds of very important studies, is through the support of our donors. So I just want to remind you again, for those of you who joined late, Sue had mentioned this, we've got the Giving Challenge coming up September 1st and 2nd, and if you would, and your friends to participate in this, and hopefully we'll raise a lot of money so that we can continue to support these very important kinds of projects. So thank you guys so much, and Dr. Steen, Dr. Kolevzon, thank you guys so much for your leadership and your work, and on behalf of all the patients, we are very grateful for all of the research teams that are working to help advance Phelan-McDermid syndrome research. And we wish you guys the very best of luck as this project launches.
Thank you.
Thank you very much.
Okay. Thanks, everybody. Take care. Bye-bye.
Bye-bye.